Hello children, how are you? Fine? Hope you are enjoying my class. So today we shall be continuing our same topic, principles of inheritance and variation, and under this monohybrid cross, that is Mendelian principle we are discussing. So in my previous class I have told you that from monohybrid cross experiment of Mendel, we came to know the two principles, Mendelian principles, that is, or in the form of laws, that is law of dominance and law of segregation. In previous class we have discussed in detail about law of dominance, how and where it is applicable, and today we shall be talking to you about the law of segregation. Law So, for getting a clear understanding about the law of segregation, we shall have to again go for monohybrid cross. Let me show it again. Tall plant with crossbred with dwarf plant. Gamete formation, capital T, small t, F1 hybrid, crossbreeding, capital T, small t, tall. Gamete of F1 hybrid, F1 progeny. If we use this F1 progeny by putting them in Punnett square, or say in the form of this, say this is when the F1 progeny will form gamete. What type of gamete they will form? One with capital T, other one with small t. So suppose this is the F1 gamete, capital T, small t, considered as a male gamete. Another plant, same F1 hybrid, having capital T, small t combination, producing gamete. So we are denoting this is a female plant, this is a male plant, male gamete, female gamete. Same capital T, small t. This is what you call Punnett square. So capital T, T. This one, capital T, small t, capital T, small t, small t, small t. In F2, we are getting phenotypically 3:1 ratio. That means in F1, try to understand, in F1 all the progenies were tall. So let me summarize. In F1 all are tall plant. But in F2, tall is to dwarf, ratio is 3:1. Of course, phenotypic ratio. Now question is, where from the dwarf plant came? It is because the F1 hybrid, this F1 hybrid, if you look here, this F1 hybrid which was tall, apparently, that is phenotypically, during the formation of gamete, the two different alleles separate from each other. That means the two alleles which were present in the F1 hybrid, this is the F1 hybrid, the two alleles which remain side by side, they did not mix with each other, and during gamete formation, the two alleles separate from each other, that is segregate from each other. As a result of this segregation, we got here in F2 generation, this is the product of F2. In F2 generation, we have seen tall plant as well as dwarf plant. So, when in F1 generation all the plants were tall, in F2 generation we got tall plant as well as dwarf plant. Where from it has come? It is because the F1 progeny, try to understand very carefully, the F1 progeny, the allelic genes present in the F1 hybrid, they did not mix with each other, and during gamete formation, they completely separate from each other. One type of allele enter to one gamete, another allele enter to other gamete. This is what we call as law of segregation. So, Mendel from its first monohybrid cross experiment gave this law of segregation. That means, so if I write the law in the form of this, so what is law of segregation? Let me make a place here. Law of segregation. This states that the different alleles which remain together in F1 hybrid when undergo gamete formation separate from each other. Separate from each other and individual allele enters to individual gamete. In F1 hybrid, although the two alleles capital T, small t, it is giving rise a tall plant by virtue of law of dominance, but during gamete formation, these two alleles separate from each other and enters to the individual gamete. That is what I have written here. This law states that the different alleles which remain together in the F1 hybrid when undergo gamete formation separate from each other and individual allele enters to individual gamete. Is that very clear to you? Yes. So let us now, by the time we came to know the Mendelian monohybrid cross, its different aspect and the outcome of Mendelian monohybrid cross, and from where Mendel came to the principles which today we know in the form of law, that is law of dominance and law of segregation. Now, Is there any violation of this principle? Is there any deviation, or always the same principles are being followed, that is dominance, especially we mean to say here the law of dominance. Means when a pair of contrasting characters are crossbreeded in F1 generation, whether all the dominant character will only express or some sort of deviation is also there. Let us see, after Mendelian monohybrid cross experiment and giving the principles of inheritance in the form of dominance or dominance and recessive, later on so many experiments were conducted. One such experiment conducted by one scientist, Carl Corey, on 4 o'clock plant, what is scientifically known, Mirabilis jalapa. Mirabilis jalapa. Another plant also he has conducted the experiment, dog plant, snapdragon, otherwise commonly called dog plant. And the same experiment was also conducted on animal also. One type of fowl, the same experiment was conducted. So let us give that experiment and we will see what is the outcome of this experiment, so that we can say whether there is any deviation from Mendelian dominance principle or it is always universal. Let's see. So let me wrap this part so that we can go for the principles apart from Mendel's law, whatever he formulated. So what was the Cody's experiment? He took snapdragon or Mirabilis jalapa 4 o'clock plant or dog plant, whatever you say Mirabilis 4 o'clock plant, 4 o'clock plant, Mirabilis. Mirabilis jalapa. Very common plant, everywhere you can find in your garden, school garden, or in your own garden at your house. You can find the same plant. Very common plant. 4 o'clock Mirabilis jalapa. What was the experiment? Because Corey, he has taken a pair of contrasting characters for conduction of same monohybrid cross done by Mendel. What was the character? He took plant with red flower and white flower. Some of the plant which is bearing red flower and some of the plant which is bearing white flower. Such types of one red flower giving plant and another white flower giving plant he has considered in his experiment. So red flower, if we denote by capital R, capital R, in terms of in the language of Mendel, the two factors governing the expression of redness. The red color of flower, that is capital R, capital R. Today what you call as gene or allelic genes. White, the recessive, this is the capital alphabet, this is the small one. White we express in this way. So when this is the parent plant, so the parent plant, the scientist Carl Corey has taken Mirabilis jalapa, the parent plant, one such plant giving red flower, pure line red, pure line was tested through several generations. Another plant was giving white flower. Now, when the parent plant forming gametes, all the gametes form contain capital R. Here, white flower, all the gametes contain small r. Is it very clear? Now, when the gamete of capital R and small r were crossbreeded, the resultant product, small capital R, small r, this is the F1 hybrid plant. Hybrid plant. Either snapdragon or mirabilis jonata. For his surprise, as per Mendelian concept, all the F1 hybrid would have been a red flower containing plant, means the plant would have given red flower only because of capital R is dominant. But here, what the scientist saw in his observation when the seeds were grown F1 plant, all the flowers F1 are pink. Neither it is red, it is white. F1 plants, neither it is red color which is dominant, nor it is white color which is missing. Whereas it became a intermediate character. When we mix red color with white color for coloring the tissues, it gives a pink appearance. The same thing he saw in his observation. All the F1 plants are giving pink flower. He is surprised. When Mendel's experiment was supposed to give, as per Mendel's law of dominance, it was supposed to give all a red flower, but it is neither giving red nor giving pink. Then he took the same plant for F2 generation. What was the product of F2 generation? Let us see. As per his experiment and observation. So make a fillet square. So F1 plant, when F1 hybrid, that is capital R, small r, by phenotypic observation pink. When this pink plant F1 hybrid were undergo gamete formation, same Mendelian law of segregation applied here. So capital R raised to one gamete, small r raised to another gamete. Now, when these gametes of F1 hybrid are taken in a Punnett square for getting the breeding experiment, let me make a little big here, the Punnett square. So what was the gamete? Capital R, small r. Here also capital R, small r. This is for female gamete, this is for male gamete. Okay, is that clear? So now crossbreeding. Capital R, progeny is capital R, small r. Capital R, again this capital R, small r, this capital R, small r, this small r means small r. Is it very clear to you? So, what is the ratio? ratio. This is the F2 generation. F2. So in F2 generation, F2 phenotypic ratio what is the phenotypic ratio? This capital R small r is 2 R capital, 1 is capital R, 2 R capital R small r, this is 1, this is 2 capital R small r, and 1 is small r. What is this? The observable property he saw, that is red, this is pink, this is white. So in F2 generation, when the gamete of F1 were crossbreeded, he saw the plants which was in F1 generation all are pink. Now in F2 generation again red and white recovered. Out of 4 sampling, he got 1 red, 2 pink and 1 white. The F1 generation where red or white both were missing. All the plants are pink. But in F2 generation again red and white both recovered. And pink also formed. In the ratio of 1 is to 2 is to 1. This one is red, pink and white. This one is white. Is that very clear to you? So, what was the difference of Mendelian inheritance and this is what we call non-Mendelian inheritance other than Mendel? We shall come to the difference. Now, before we tell the difference, so what happened? Why? The F1 hybrid is not giving a character like Mendel's principle, red, or why the F2 generation separated. F2 generation, the phenotypic ratio we got in the form of 1:2:1 means 1 red, 2 pink and 1 white. That means where from this pink flower came, or why all the F1 plants are pink, F1 hybrid. It is because here the red is not completely able to dominate the white allele. Neither the white allele is totally recessive. That means none of the 2 alleles are behaving like a dominant or recessive. As a result of which they are producing a blending of these 2 characters, that is pink. We can express it, we can explain it in the form of the enzyme production also, but there is no point of discussing here. Means actually all the genes are responsible for producing protein, these enzyme protein which are acting on the substrate to give a product. Here the dominant or the recessive alleles in terms of production of gene, that is active gene produced, that is called functional gene produced, or less effective gene produced, or no gene produced at all. We can explain in terms of this also. But you try to understand that here the red allele or the color for expression of red character, that is capital R, and the color for expression of small r, that is the recessive gene, they are not behaving as a true dominant or true recessive. When they come together, they produces a sharing, blending, mixing of character. This is what we got it in the form of pink color. But in F2 generation, again red and pink recover. Why? Because here the ratio of one red, two pink and one white. Why? Because here both allelic genes are capital R, capital R. No other genes are there. That is why it is producing red. These two genes both are responsible for production of red color. Here again these two are pink. Why? Because they have capital R, small r. That means again they have two different allelic genes and they are not behaving in the form of completely dominating the recessive gene or recessive gene not completely masked by the dominant gene. They are sharing their blending character. That is why you got two pink color containing flower. And one is perfectly white because both the allelic genes are responsible for carrying the white. This is what we call non-Mendelian inheritance. These are the exceptions, experimental outcome of the crossbreeding of Mirabilis java plant, or same thing was done when snapdragon dog flower was taken into consideration. Again, when the same experiment was tried on animal, one kind of fowl was very beautifully observed, black color fowl with white color. These two color fowl when were taken, say that black color governed by capital B gene, white color governed by small b gene. When gamete was produced, these are the parental combination, gamete, capital B, gamete, small b. When these gametes were crossbreeded, F1 hybrid was produced, capital B, small b. If we think about the Mendelian concept, then the B, capital B would have been a dominant character, hiding the, masking the effect of small b. So the resultant fowl, whatever the chicks so produced, would have been a black color. All were having black color. But no, neither it is black nor it is white. It became blue color. Blue. New color appears in F1 hybrid. So again, this is a violation of what we got in terms of Mendelian dominance, law of dominance. That means the principle of dominance here is not applicable. Here, the capital B and small b are sharing equally, giving a blending of character, color, that is a blue. Is that clear? Now, when this F1 progeny was undergo gamete formation, F1 gamete, it produced capital B, small b, as per same law of segregation of Mendel. Now, when this, if I rub this part, if I rub little here, make some space, Now when the F1 gamete of chick, capital B, small b, this is capital B, small b, this is male, this is female gamete, where crossbreeded, capital B, small b, capital B, small b, and this small b, small b. What is the resultant progeny? Resultant progeny, that is black F2. In F2 we are getting now black out of 4, 1. Blue out of 4, 1 and 1, 2. And white out of 4, 1. So again, the color, parental color, black and white, what was missing in the F1 progeny, again recovered. And we see here in the form of 1:2:1 ratio in the F2 generation. That means in F1 hybrid, where the dominant and recessive alleles in terms of Mendelian concept, which assemble together, but they are not able to express themselves in the form of dominant and recessive, a blending character will work, and in F2 generation, again they separate, following the law of segregation. So this is what we call a non-Mendelian inheritance. But can you tell me one difference between Mendelian and non-Mendelian inheritance? If you notice carefully, in Mendelian inheritance, if I write here, Mendelian inheritance, the phenotype of F2 3:1, genotype of F2 again different, that is 1:2:1. So in Mendelian inheritance we have seen the F2 generation of monohybrid cross, it gave a ratio of 3:1, but genotype is 1:2:1. But in case of non-Mendelian inheritance, both the phenotype and genotype in F2 are same. Here the phenotype is 1 red, 2 pink, 1 white, and the genotype is also 1 red, 2 pink, 1 white. So in non-Mendelian inheritance, both phenotype and genotype of F2 generation are exactly same in the form of 1:2:1 ratio. But in Mendelian principle, the phenotype of F2 generation is 3:1, whereas genotype is 1:2:1. These types of questions you will certainly get in your board examination, or even many times it has come in your NEET examinations, medical entrance test. That what is the basic difference of the phenotypic and genotypic ratio of Mendelian inheritance and non-Mendelian inheritance. In Mendelian inheritance, we see the phenotypic ratio of F2 generation of monohybrid cross is 3:1, whereas genotypic ratio is 1:2:1. On the other hand, in non-Mendelian inheritance, both the phenotypic and genotypic ratio of monohybrid cross of F2 generation is 1:2:1. Is that very clear? Is it very clear to you? Okay. Till then, thank you.